Everybody. Due to COVID-19, we are at home and I want that your studies will not suffer. That's why I'm starting from the where I left. I left at Unit 3. Now today I am starting the viral diseases of plants, that is Unit 4. Today we are first taking the viral disease of plants, that is the yellow vein mosaic of bhindi. The yellow vein mosaic of bhindi is the same as that of late blight of potato, which we have done. In late of blight of potato, we have done is, this is the leaf and the symptoms started at the tips and then in the margins. And ultimately it spread to the whole leaf and it results in the chlorosis. And chlorosis is, as you know, is followed by necrosis and then death of plant, right? So in yellow vein mosaic, it is a viral disease. In viral diseases particularly, it is different from the fungal diseases. Here the vein clearing will start. So vein clearing, what is vein clearing? Vein clearing is basically the process in which the leaves has a veins, and these veins, the leaves has a veins and like these veins, and the clearing starts here. When clearing start at the vein point, then it is called intravenal. What it is called? Intra. When it started here, it gives green, yellow, and this green, yellow is called mosaic, right? And as first is the, we have the venal clearing, that is intra, which is followed by the inter. Then venal chlorosis, because of venal chlorosis, the leaves show mosaic, that is green, yellow, and then the veins and veinlets, they become thick. And this results in yellowing of leaves. And as we know, as the leaves they yellow, then photosynthesis decreases and respiration increases, and which results in chlorosis is followed by the necrosis, and that results in the defoliation of the leaves, and ultimately then the fruits which are formed, because photosynthesis is not there, fruits are dwarfed, malformed, yellowish green in color. And as it is in bhindi botanical name is hibiscus, that is virus is hibiscus virus or yellow vein mosaic virus. It is a small and spherical virus, and it is basically transmitted, not the sap transmitted like we have read in the BSc 1, that is tobacco mosaic virus. It is sap transmitted, but it is not sap transmitted, but basically it is transmitted by the white fly, jisko we tell Bemisia tabaci. And besides that, it is grafting, right? And the several hosts are there, croton, malvestrum, ageratum. And you know, by these disease, what are these control? First, it is the destruction of the weed host. Then every time when I will teach you the viral disease, Bemisia tabaci, white fly will come, then the spray of metacystox, ecatox and rogor, it will come. Like we have read in the Botex mixture in the fungi. And other is the resistant variety, that is Pusa savani. And this is the what we have, the yellow vein mosaic. So yellow vein mosaic, I will abstract it. Yellow vein mosaic is basically the disease in which venal clearing, venal chlorosis, yellow network of veins, first intravenal, then intravenal, and then complete yellowing of the leaves, the stunted growth because respiration is stopped, nearly stopped, fruits are dwarfed, malformed and yellowish green in color, right? Now next we will take the second disease. Welcome back everybody, and the next disease is the Tunguru disease of rice. Tunguru disease of rice is basically a very important disease. So symptoms are show mottling. What is mottling? As I told you, in virus we have mosaic. Mosaic means a leaf. At a bhindi mosaic we have done. In bhindi yellow vein mosaic, that is veins yellow, then this part remains green, yellow, and when the green yellow it comes, it is called mosaic. Here it is not green yellow, it is yellow orange discoloration. Yellow orange, and because of that, the photosynthesis decreases, and as respiration increases, we have first chlorosis. Chlorosis is followed by the necrosis, and the necrosis, and we have the stunted growth and the panicles. As we know, the rice inflorescence is the panicle, and the panicles which are developed because photosynthesis is low. There is no source of energy. So panicles, where the grains of rice are there, they are very small, and sometimes there are no grains. Or in early infection, the young plants may dry prematurely before they flowers. The causal agents are rice tungro bacilliform virus, RTBV. It is double-stranded DNA virus. We will get this question in UGC. And rice tungro spherical virus, RTSC. It is single-stranded. This we get question in the UGC. In both it will come. We have to take which is double-stranded and which is single-stranded. Transmission is not by sap, as TMV. I already told you. It is by grasshopper called Nephthyrix nigropictus. And this basically transmitted through the grasshopper. And when we go for the control, the transgenic japonica rice and basmati 370 are basically raised. And besides this, some mites infected plants, they show resistance. So my early sowing is the best control to this disease. So we will say tungro is showing a mottling, then stunted growth, small and sterile panicles, early infection, the whole plant dry prematurely, and the causal organism is RTBV and RTSV. RTBV is double-stranded and RTSV is single-stranded. Not transmitted by sap, transmitted by grass leaf hooper, and the control is use of transgenic japonica rice and basmati 370. Besides this, the plants which are infected with the mites, they show resistance to the disease, and early sowing is the best control to this disease. Morning everybody. Now I continue again the second disease, that is the bunchy disease of banana. The bunchy disease of banana is like that. We have a plant, and in plant the leaves, they bunch, that is name from the bunching, and they are erect. They bunched and erect. In bunchy disease, the leaves are bunched together at the apex to form a rosette called bunchy top, and this is called rosette. And the bunchy top plants are stunted, as they show erect leaves. As I told you, when the leaves are erect, so when sun rays will come, so photosynthesis will reduce because they are erect. For photosynthesis, they should be slanted. They are erect. The photosynthesis is reduced. Respiration is increased because photosynthesis is inversely proportional to the respiration, and that is why the energy is not produced, and it shows the stunted growth. The margin of leaves are wavy. And because the photosynthesis is not there, the fruits which are formed, they are small. They are small, dwarfed, malformed, and because they are small, the yield is less. The causal virus is the banana bunchy top virus as the disease. This is also called banana virus 1. It is a single-stranded RNA with a isometric symmetry. Icosahedral means it has 10 faces. It has an 80 to 20 nm in diameter. And the transmission part is the chief insect vector, that is Pantonolia nigronervosa. And the other part for the transmission are the suckers. And basically this disease is restricted in the phloem tissues. And the other alternate hosts are the Musa fehi and Musa textilis. So what we came to know, that the bunchy top, the leaves, they bunch together and they form a rosette, stunted growth, malformed leaves, and that caused by the banana virus 1. And when we are coming to the control, we know na rahega bans, na bajegi bansuri, the chief insect vector to be removed. So spray of kerosene and parathion, as in Bemisia tabaci, we spray Rogor, Metacystox, but in case of Pantonolia, we spray kerosene or parathion. And after that, as it transfers to the suckers, so seed certification is required for sucker plantation, that in that sucker disease is not there. Then other is the we have the biological control of the aphid vectors through the parasitic wasp. Now I will continue again for the next disease short while. Okay, welcome back everybody. And the third disease is the leaf curl of tomato. So as I already told you when we do the fungal disease in the class, like this is the leaf. The leaf curl, as you know, this leaf curls inside, and when it curls inside, then there is a sound called crinkling sound, and besides when it is curl inside, the photosynthesis surface area is reduced, and when the photosynthesis surface area is reduced, then respiration increased, and this results in the first the chlorosis, you know, like this chlorosis followed by necrosis, and then because necrosis, the plant becomes stunted, and due to the shortening of internodes, and the whole plant look pale and appear bushy. As you know, curling, rolling and crinkling of leaves and chlorosis, necrosis, stunted growth, and we came to know that fruits are basically it is a complete sterile plant. Fruits are deformed, small, and it is caused by the tobacco leaf curl virus, and it is a seed borne, and it is caused by the whitefly Bemisia tabaci. As we know, as we done in the Bindi mosaic, as soon as Bemisia tabaci came, the spraying of Ecatox, Ragor, Parathion, it will come, and definitely it will come. And besides it, we have a one resistant variety called Lycopersicon peruvianum. Tomato botanical name is Lycopersicon, and then is peruvianum, and show a high degree of persicum. So a high degree of resistance. Besides this, the growth regulator like GA, the gibberellic acid, 2,4-D, which are also used to reduce the disease and increase the yield. So in crux, we will say it is the curling of the leaves which it reduces the photosynthesis. As we know, the photosynthesis is inversely to respiration. Respiration increases, and that result in the chlorosis of that area. Chlorosis is followed by necrosis, and ultimately necrosis is followed by the plant death. So plant death or deformed fruits, because here we are talking about the tomato and the main concern is the fruit. So deformed fruit means less yield, and the less yield means it is the farmers who are the main. Welcome back, and the next disease is Distigia of citrus. The common symptoms of disease is change of color of leaves from bronze to yellow, as we done in the Bindi mosaic. That is the intravenal, and then is the intravenal. From here the leaves change to yellow. Yellow means chlorosis. As you know, the chlorosis every time follow the necrosis, and necrosis means then leaf fall, the defoliation of leaves. Besides this, the most characteristic feature of this is longitudinal grooves. Longitudinal grooves in the stem called stem pitting, and because chlorosis is there, the photosynthesis is less and respiration is more, means stunted growth. And as it is a citrus, so main concern is the fruits. So fruits are small and distorted. Seedlings, when attacked, develop chlorotic seedlings in the early state. It is a premature death of the seedlings. The causal agent is the citrus tristigias virus. It is a single stranded. It is done by the grafting and by the vector called Taxoptera. It is also transmitted by the dodder. And the control is to Narega Bans, Nabajagi Bansari, that is remove the susceptible trees and grow the resistant plants. And then is cross protection. Sometimes the trees are inoculated with mild strain of virus, and this strain does not allow the virus strain of this citrus tristigia to come and give the resistance to the plant. Besides that, it is grafting, right? And the several hosts are there: Croton, Malvestrum, Ageratum. And you know, by these disease, what are these control? First, Narega Bans, Nabajagi Bansari, it is the destruction of the weed host. Then every time when I will teach you the viral disease, Bemisia tabaci, whitefly will come, then the spray of Metacystox, Ecatox and Rogor, it will come. Like we have read in the Botex mixture in the fungi. And other is the resistant variety, that is Pusa savani. And this is the what we have, the yellow vein mosaic. So yellow vein mosaic, I will abstract it. Yellow vein mosaic is basically the disease in which venal clearing, venal chlorosis, yellow network of veins. First intravenal, then intravenal, and then complete yellowing of the leaves. The stunted growth because respiration is stopped, nearly stopped. Fruits are dwarfed, malformed, and yellowish green in color. Right? Now, next we will take the second disease.